Okay, so this is the video that I would have liked to have shared with you all earlier during our class. This video is going to explain a bit about using the PCR process to help with DNA cycle sequencing. So let's say we are starting with an unknown sequence of DNA. We have a pathogen's DNA that we want to sequence. We already know that the individual afflicted with the pathogen is a Homo sapien, a human. So we really don't care about the DNA that belongs to the human. So essentially what's going to happen is we get a DNA sample from this individual, and anything that pops up as human DNA is irrelevant, but anything that's considered foreign DNA would be of interest to us. And so initially you have DNA in a little tube that would be of an unknown type. Now you have to have some speculation as to what type of DNA you're dealing with. So let's just say we thought that Sue Smith was ill with the flu. So knowing the DNA sequence of the typical flu virus would be a necessity in order to select the correct primers for this process to go down. Every flu virus is going to be a little bit different from the others, but they do have some DNA that's common to all. In fact, there's quite a bit of DNA that's common to all. That's the case with every organism. And so in that case, we have a region of DNA that would be highly conserved, meaning it's the same from one type of flu virus to the next. So we would take a primer that we know would match to the known portion of the flu virus DNA. And so let's just say that that's this right here. This is a primer manufactured in a lab. So what's going to happen is originally your DNA should be double-stranded. For the sake of time, I just made a single strand of DNA here, but there would be a complementary strand. What happens next is this sample is in a tube, and we want to make many copies of it. So the process of PCR would take your DNA and it would heat your tube up to a very high temperature, a near boiling temp, in fact. And what that does is it denatures the DNA. So that means the DNA strand unwinds and unzips down the center of the strand where the two DNA pieces or strands are woven together and held together by a hydrogen bond. So essentially we have two strands of DNA. We're going to deal with just one of them right now. The next piece, once the DNA strands have separated, is that we take the temperature down about 20 degrees Celsius, and that allows a primer that you've added to the mix to attach or bind to complementary base pairs. So if we have a T exposed on one of our strands, we'll have the A portion of our primer that would bond to it. The A exposed here would bond to a T, and so on. So this little primer right here was predetermined. In addition to having one that should attach to, let's say, this end of our unknown DNA strand, we would also have a second primer downstream, down that way, at the very end of what would be a very long strand of DNA, and it would be attaching to its opposite end as well. Basically, your primer is bracketing the gene that is unique to the individual species that you are sequencing the DNA for. So let's just say we think it's the flu virus. We've attached primers at the lower temperature. They theoretically are attaching inside of a little tube in a machine. And if all goes well, we're going to have successful copies of that DNA made. Later, we're going to read that DNA and we might find that we get no results. And why did we get no results? Because perhaps what ends up happening is this DNA did not actually belong to the flu. So what might have happened is your primers that you suspected would work with your flu virus won't have anything to attach to if in fact what's in the tube is not flu virus. Let's say Sue's actually sick with something else. So your primers that you suspect might attach. Let's just say we suspect something other than the flu. We've put primers in place specific to that suspicion, and they actually are attaching in the tube inside of our machine. Okay, so now what happens? A DNA polymerase enzyme comes along. So if I have something small I can use as an enzyme, let's just say a small little wadded up piece of paper that I happen to have here, okay? this little enzyme is going to come along and it's going to grab hold of nucleotides from a collection of free nucleotides that we have in our tube as well. So let's just say this is my tube and I have all these free nucleotides floating around. This little DNA polymerase is going to come grab them at random. And what it's going to do is it's going to build the remainder of this strand of DNA right here by matching all the corresponding nucleotides together, A's with T's and G's with C's. So from the point of the primer onward, if it's doing things correctly, which it usually does, it should be placing an A next. Then it would be placing a T, then a C. Okay, I think you're getting the idea. A G would follow. And let's say it places another C. I'm gonna go one more. And then it places an A. And it just so happens that the A it's grabbed hold of is special. That particular adenine nucleotide is fluorescent. It has a special fluorescent dye that's been added to it chemically. And what that now does is it tells this DNA polymerase, and now you're done. There should be no more building of this DNA strand. I don't know exactly what happens there, but it's a conformational change with some sort of a chemical interaction where the DNA polymerase can no longer build any remaining nucleotides onto our second strand of DNA. So essentially what's happened is our double-stranded DNA has been truncated at this point. The fact that there's excess DNA here is sort of irrelevant. All that matters is we have a double-stranded set of DNA right here, okay? Now, this is happening multiple times over. So inside of your tube, you had a second side to this DNA strand right here. The exact same thing is happening to that second strand of DNA. I didn't make one, so I can't exactly show you, but the second strand of DNA is going to actually be built in the other direction. So the second strand of DNA would have had its primer on this end and would have been building in this direction. So long story short, we're gonna end up with varying lengths of DNA that's made. Now, you don't just have one single piece of DNA in PCR, you're making lots of this. So this strand got truncated, but now this strand is gonna become separated when the temperature is heated up. So once again, these two will denature, get pulled apart from one another, and now the process starts over. The primers that are available in the mix will come on over and attach, and then the remainder of the strand is going to be built by the DNA polymerase. And as it moves down the length, it's grabbing random nucleotides to fill in all of the spaces as it builds, yada, yada, yada. It's doing what it should be. It actually proofreads to make sure it doesn't make mistakes. And lo and behold, look what happened. It just so happened to grab a fluorescent thymine right here. So once again, that's going to truncate the double-stranded DNA right at that point, okay? And then it will go through the process of heating the sample. These two strands of DNA will become separated, and we're going to start to see that yet again, the strands primers attach, the nucleotides are going to be added to place by the polymerase doing its job, and so on and so forth. So theoretically, if in the mixture we had enough primer, if we had enough free nucleotides, and if we had enough fluorescent nucleotides, what you basically end up with is more or less a sequence of the DNA that you would have been a complement to this original strand. So what I had done right here, for example, I had a strand that ended with the letter T. I believe I had a couple A's, sorry about that, a couple A's. And then we had a letter T that was fluorescent. The first strand that I built in this video had the primer, and then, excuse me, it had two A's. Oops, this is difficult. These little pieces want to stick. And the T which in this case isn't fluorescent. And then let me revisit what the order should have been. A C, and then a G, and then it had a C, and then we went, whoops, fluorescent A. So imagine doing this enough times that every single one of the letters from the point of the primer onward at some point is a fluorescent letter. If you did this enough, you would have a fluorescent A at some point, then a fluorescent T at some point, which we already had, then a fluorescent C at some point, a G at some point, a C at some point, and now we have the A, and so on, all the way down the line. So now you have this mixture in your tube of all this stuff, right? What do you do next? Now you put all of this into a gel electrophoresis, more or less. It's a machine that's going to do the exact same thing. And what happens in a gel, if you recall from previous learning, is the DNA will move through a gel lattice structure, but the smallest fragments always move faster. They don't get stuck as much in the gel. So if you have a fragment that is very little, let's say that little, that might come through early on in the gel electrophoresis. In fact, the one that's going to come through first is the one that's just one single nucleotide long. So our A comes through first. If it's fluorescent, it will be read by the laser. The next letters to come through would be a sequence of A followed by T, which I cut somewhere. Here it is. Okay, that one would come through next based on its size. And if the T was fluorescent, it would be read by the laser. So if I kind of edit these in the moment, I'm just going to put a little mark because I can't let go of my phone. Okay, that's a fluorescent A. That's a fluorescent T. Now we have a fluorescent C. The machine is reading these fluorescent letters and it's spitting them out as a graph of humps of certain color. And that color indicates whether it's an A, a T, a C, or a G. So again, if I kept going, the next piece that would come through based on size alone would be this one. And let's say its G was fluorescent. That's why it only ended up being this long. Once the fluorescence was selected, the rest of the build did not occur. So continuing on, the next one would be this guy, and the C would be fluorescent. Continuing on, the next one would be this one, and the A would have been fluorescent. Okay, so if we take all of this and we start paying attention to what the fluorescence is, we start with an A. The next fluorescent was a T, then a C, a G, a C, an A. And what we're really doing here is we're crafting the sequence of letters that correspond to our original DNA strand sequence. So while I haven't done the entire thing, look at that correspondence there. A with the T, T with the A, C with the G, G with the C, C with the G, A with the T, and so on all the way down the line. So again, theoretically, if there's enough in the mix, you should be able to spit out a series of laser red fluorescent nucleotides that are interpreted and put into the correct order so that they become the complement to this first strand of our DNA that we see right here. Okay, hopefully that makes sense a little bit better than what we did in class today. Thank you for your patience.